So to start, I'm just going to review a little bit. We already talked about these things, and yesterday we did a bunch of things to kind of reinforce what we know about DNA. So DNA is a nucleic acid, deoxyribonucleic acid. And we made models of DNA. Right? We did a packet about DNA. You did the gizmo, and you looked at how DNA replicates. So you should have a pretty good understanding now about DNA. Remember, it's made of a bunch of nucleotides, all bonded together. A nucleotide has three parts: the phosphate, the sugar, and then one of the bases. Remember, there's four bases in DNA: A, T, G, and C. Adenine, thymine, guanine, cytosine. And they pair up. And you did in the activity, we're matching the bases. Apples and trees, cars and garages. A and T are complementary to each other. When you have an A on one strand of the DNA, its opposite side has a T. And C and G go together. Guanine and cytosine, same thing. And we also know that when cells are reproducing, the DNA needs to copy, right? We need to make a copy of it to put it into each of the new cells. And that's called DNA replication. And that's what you did in the gizmo. Remember, the hydrogen bonds between the two strands separate. An enzyme helps this to happen. Then new nucleotides attach on each side. And then eventually, after that happens through the whole strand of DNA, what you end up with are two identical copies of the DNA. They're all exactly the same. And so the DNA has replicated as copies. So that's how DNA replicates. Here's some just diagrams. Remember, the shape is a double helix. DNA has two strands, two sides to it that sort of curl around each other. Okay. We see the hydrogen bonds that hold together these two halves of the DNA strand. Now, today, we're going to start to talk about a different type of nucleic acid. So we have DNA. There's also this other type of nucleic acid called RNA. RNA is also found in cells, in the ribosomes, in the cytoplasm, in the nucleus. And it's similar to DNA, but there's some important differences that we'll talk about. Okay, so first off, RNA, like DNA, it's a large molecule. Like DNA, it's made of nucleotides, which have a phosphate, a sugar, and a base. The sugar is a little bit different, though. The sugar that was in DNA was called deoxyribose. The sugar that's found in RNA is just called the regular ribose. And so RNA stands for ribonucleic acid.
So it has protein in it?
No, it's not. It's
not like ribosomes
ribose, not ribosome, but we're going to talk about ribosomes in a minute. So we have phosphate group, then we have the sugar, which is ribose, and then we have our bases. And you'll notice the bases are similar: cytosine, guanine, adenine, but there is a difference. There is a base called uracil. It's in place of the base thymine. So there's no T in RNA, but there's a U instead. U for uracil. We also notice RNA only has one side to it, one strand. You can see it's here, it has the backbone, and then it has the bases sticking out of it. So that's a little different also. And RNA has a different job to do. Okay. There's three main types of RNA. And each of them has a specific job to do. And we'll talk about that as we talk about protein synthesis. So we put a little letter in front of the different types of RNA. Okay. mRNA, that stands for messenger RNA. And we'll talk about what each of these things does in a minute. So mRNA is messenger RNA. Its job is to take a recipe from the DNA in the nucleus and bring it out of the nucleus to a ribosome. We have tRNA. The T stands for transfer. The job of transfer RNA is to carry amino acids to the ribosome to be connected together. And then the third type of RNA is called rRNA, and that stands for ribosome RNA. Ribosomes are actually made of RNA. And all three of these types of RNA work together to help cells build proteins. Call that protein synthesis. Synthesis means to build up a larger molecule. So if we think about DNA and RNA, we could compare them. They have some important differences, right? DNA has 2 strands. It's a double helix. RNA has 1 strand. It's just a single spiral, a helix. The sugar in each one is different. In DNA, it's deoxyribose. In RNA, it's ribose. The nitrogen bases are similar, but with a slight difference. DNA has A, C, G, and T. RNA has A, C, G, and U instead of T. They're both made of nucleotides, and they both have basically the same components. So what our focus is on something that's called protein synthesis. So let's think back to what we learned about proteins. They are large molecules. Proteins are very large molecules. And they're made of many different amino acids strung together. There's 20 different amino acids. And they can be put together in huge combination, in many combinations, to form different proteins. The way those proteins get built, the recipe, the instructions for how to make these proteins, which amino acids to put together, that information is stored in the DNA. When we use the word genes, we say we have a gene for blue eyes. When we say a gene, what we mean is that in the DNA, there's a recipe for how to build a protein that gives our eyes their blue color. A gene is just a recipe for building a protein. And that's what our genetic code is. In the DNA, the order of those bases, the A's and G's and C's and T's, the order of those, that's our genetic code. If you looked at all my chromosomes, all my DNA, the order of the bases is going to be slightly different from yours. And that's why you and I have different traits and characteristics. If I were to compare my genetic code to my sister's, it would be
Kind of the same.
more similar. Not the same,
Kind of.
but more similar because we have the same parents. So we have lots of the same genes. The sequence of those bases would be very similar. We have about 3 billion of those bases make our genetic code. So you're looking at all those A's and T's and G's and C's. Your genetic code is about 3 billion of them. That's what makes you you. And the codes that are stored in those DNA strands are needed to make the proteins that make our body. Right? We're made of proteins. Enzymes are made of proteins. Hormones are made of proteins. Antibodies are made of proteins. Proteins are almost all of how our body works. And so the instructions to build those proteins are really important. So the way they're made is through a combination of work between the DNA, which basically stores the recipe, has the original version. And that original recipe is used to create the proteins by RNA. So here are the basic steps. First, when one of the recipes in the DNA is going to be turned into a protein, there's several things that happen. First off, the DNA has to unzip. And the gene, the sequence of bases in the DNA, the A's, T's, G's, and C's, is copied into a different form. It's copied onto an RNA molecule, a messenger RNA molecule. That happens in the nucleus. DNA is stuck in the nucleus. It can't leave. It's too large of a molecule. But if that recipe that's in the DNA gets copied to messenger RNA, that's a smaller molecule. It is able to leave the nucleus and go out into the cytoplasm of the cell. So the DNA gene is copied first to a messenger RNA molecule. And that messenger RNA heads out of the nucleus. It's carrying the recipe to the place where the protein is going to be actually put together. Hopefully you remember from when we studied cells, it's ribosomes that do that. Ribosomes are what put together the amino acids to make a protein. So this messenger RNA that has the instructions goes to the ribosome. It's also made of RNA. To bring the instructions there, to tell the ribosome how to start creating this protein. And then the third type of RNA, which is called transfer RNA, starts bringing in the correct amino acids. Remember, there's 20 different amino acids that could go into this protein. And so transfer RNAs start bringing in the correct amino acid, one after the other. And the ribosome links them together, one after the other, thousands of times to build a protein. And eventually, the protein is completely formed. It has all the amino acids stuck together, and then it can be released from the ribosome and do whatever that protein does. Could be a protein that makes up muscle. It could be a protein that's a hormone. It could be a pigment protein in our skin. It could be many different things. But that's the basics of how the instructions to build a protein that are stored in the DNA actually get turned into a protein in a cell. So we can look at a bunch of diagrams. We're going to have several videos to show this visually. It has two parts. The first part happens in the nucleus. This is so again, this is DNA, this double strand. It unzips, exposing one of the genes. And from that, a strand of messenger RNA is created. It's complementary. Just like the bases in DNA match up, the bases in RNA also match up. I know you have this, but don't look at it for a minute. So, if the DNA gene is this, G, G, C, A, A, T, A, C, C, G, A, T, what is this messenger RNA that's created? What are its bases going to be? If the DNA has a G, what do you think the RNA will have?
RNA will have something different. C.
C. This G would have
A T. C.
C. A or C would have
A G,
A.
T,
Oh, wait a minute. RNA doesn't have a T. It's got a U instead.
oh, sorry.
That's the only difference. So yeah, that would be G goes to C, G goes to C, C goes to G. A now matches with U because RNA doesn't have T. It doesn't have thymine, it has uracil. So anywhere you had A and you would have a T, instead it's a U. And so that's the sequence you have. So we have DNA, the recipe gets copied to messenger RNA in the nucleus. And then it can head out of the nucleus, make its way through holes in the nucleus, head to a ribosome. This part is called translation. It's actually building up the protein. And the way this works is every 3 letters on the messenger RNA is a code for one of the 20 amino acids. So AAA is a code for one amino acid. AGU is a code for a different amino acid, and so forth. So the messenger RNA has the code now. Transfer RNA molecules start bringing in amino acids one after the other, and they get linked together by the ribosome. There's special codes, one that starts the protein being made, and then there's another code that says, okay, this protein is finished. So this is a diagram we often use. This is a diagram that shows us, helps us take the messenger RNA code and figure out what is it a code for? Which amino acids is it a code for? So the way this chart works, and you'll have to use this, this is often also on the Regents exam, this chart. All right, give me 3 letters, 3 bases from RNA. Any random 3. A, G,
R.
only A,
I mean, not R. J.
only A, U, C, and G.
U.
All right, A, G, U. That's 3 letters. It's a code for one of the 20 amino acids. So how do we use this chart? We take the first letter, A. We find it on the left. See how it says first base? All right, here's A. Okay. Then we look at the next letter, G. Okay, here's G. So now we go down here. It's going to be somewhere in this box. And then we look at the third letter, U. Here's U. Oh, look, there it is. AGU. AGU is the code for an amino acid called serine. I'm going to give you one to look up. GAU. Try and find it in this chart. You have a copy of it on your paper also. GAU. Nope. Raise your hand when you know what amino acid it's a code for. GAU. Jordan, what is it? Let's see. G is here. So it's going to be somewhere in this row. A. Okay. It's going to be in this box. U. There it is. GAU. Correct, Jordan. ASP. I'm going to give you another one. UGA. Find UGA in your chart. Okay, so this one's a weird one. UGA. So what is it? Stop. U G A. Yeah, so that says stop. This is a special code. Once the codes are being read at the ribosome, if it reads this code, UGA, that tells the ribosome, okay, stop. This protein's finished. All of the amino acids it needs have been connected, and the protein is finished. You could see there's a couple stop codons. There's one. Here's a couple more. UAA is a stop codon. UAG is a stop codon. So that's how you take some bases in messenger RNA and figure out which amino acids they're a code for. There's other versions of this. Sometimes there's a circle version, but the point is you find the 3 bases and you can determine which amino acids it's a code for. So, this is not very good resolution. So again, this is just another model showing you protein synthesis. It's kind of the same thing. The green is the nucleus. We could see the DNA is here. We could see it's unwound a little bit to expose this gene. And these bases have created a messenger RNA molecule that has matching bases. The messenger RNA can leave the nucleus, can head out here to the ribosome. And then every 3 bases, these are the amino acids, a new amino acid is getting put in. These transfer RNAs are bringing the correct amino acids so that it can be linked together to make the final protein. This is better resolution, same basic thing. Nucleus, DNA unwound, copied to messenger RNA, which leaves, goes to the ribosome. The transfer RNAs are bringing in the correct amino acids to form this protein. And so these diagrams are all obviously simplified. A protein actually has thousands of these amino acids, one after the other stuck together. And once they're all attached, it folds up into a very unique shape. Remember, what's important about how proteins work is their shape. Whenever you see protein, you should think shape. Hormones work because their shape matches a receptor molecule.
Okay.
Antibodies work because their shape matches an antigen.
Okay.
Protein shape, enzymes work, the proteins, because their shape matches the substrate. So all proteins, the shape is the key thing. Here's just a little animation showing the different steps. I'll wait till it restarts to explain a bit. So the first step, the DNA is unwinding, messenger RNA is being created, it went out to the ribosome, and now transfer RNAs are bringing in the correct amino acids. They're being bonded together until the protein is finished. All right, so that's protein synthesis. It's complex, but looking at the steps, it's really 4 basic things happening. So we're going to watch a couple videos here to reinforce this. Then we have an activity. You're going to do some coloring and look at this process to help make sure we
They don't appear to have any difference in change after they apply those protein amino acids